Shall we begin?
Yes, ma'am, we can get started if sir is ready.
Of course. Yeah. Thank you so much. Hi, everyone. Very good evening. Welcome to session 5 of our internship program. So far, we've studied about systematic review process in detail, right from screening to search strategy to critical appraisal to a lot of things. And today, we come to a session called network meta-analysis, which will be taken up by Dr. Rajpal Singh. He will be taking us right from the basics of what is meta-analysis to ITC to NMA, followed by a live demo of running the meta-analysis. Okay, so it's an important and exciting and interesting session. With that, I hand over the stage to Rajpal. Rajpal, all yours, please.
Thanks, Richa. So, Dr. Richa, are you able to view my screen as well as other team members?
I can see the agenda, Rajpal. Is that what you're
Okay.
showing?
Yes,
Okay.
yes.
Okay.
So let me walk you through the agenda first. So we will be discussing the stepwise approach for what happens after systematic review and how we move ahead with the network meta-analysis. Then we will discuss about feasibility assessment, which is a step which is done prior to conducting a network meta-analysis to assess that which studies should go into that analysis. And then what type of outcomes do we have, how to calculate missing values, and what are the enemy results, how they look like, and how we have to interpret them. So first, we conduct a systematic literature review that you have learned in the previous sessions. And after the completion of systematic literature reviews, we do also the quality assessment of included studies. Why we do, we will be discussing it in the later slides. And post that, there is an activity named as feasibility assessment, where we check that whether which studies or which involved clinical data should be part of a network meta-analysis. And post that activity, we actually conduct a network meta-analysis. So this is a Prisma flowchart, how a flow works in systematic literature review. After completion of the data extraction activity, we move ahead with the feasibility assessment. So why is a feasibility assessment, or how we identify studies? Basically, we construct network diagrams to understand that whether all the treatment arms of the included studies from the systematic literature review combines with each other or not. So it is generally a network diagram that we prepare manually or by using R codes as well. Then we do analysis for identification of effect modifiers. Effect modifiers are basically the baseline characteristics of the clinical trials. Suppose age is a factor. We cannot compare 5 studies that are reporting on adults with a study which is reporting on adolescents or children. So these type of effect modifiers are identified for that specific therapy area and for the NMA objective, and then we judge basically on the basis of the baseline characteristics of each clinical trial. So let me move fast to there. We check for the transitivity and consistency. So students, please take care that these terminologies, these might be new to you, but you can go through this recording again and write down the definitions of transitivity, consistency. These terms, those will be helpful in your further uh, practice as well. So transitivity, it is basically, you can say that if treatment B is hypothetically better than A and A is better than C, then we can presume that B is better than C. This is clearly a mathematical comparison that we do with the clinical data. Then we also check for the consistency. Direct and indirect comparisons must be roughly in agreement with each other. They should not be like that for a specific set of analysis, those values are not matching with the base case analysis. We also check for the heterogeneity because we have to we have to assess basically that all the clinical studies that are part of the systematic literature review, those we have extracted, should be homogeneous enough to conduct an analysis. So there are 3 types of heterogeneity. First one is the clinical heterogeneity. So it basically based upon variability in the participants, interventions, and outcomes of the clinical trials. So basically that all the participants or the inclusion criteria for the patient recruitment should be similar. That cannot be same across the clinical trials, but yes, those has been selected. It should be similar ones. Also, the interventions, it should be the intended interventions as per the scope of systematic literature review. We cannot compare basically crocin with an anti-cancer drug because the outcomes and the treatment class are very different. Then we also check for the methodological heterogeneity. Basically, it is the variability in a study design and risk of bias. Variability in study designs means ideally in an ideal scenario, observational study should not be compared with an RCT, that is the randomized clinical trial, in a network meta-analysis. Why? Because the study design is a very different for them. But in certain cases, we also do this type of analysis where we compare observational studies as well as clinical trials. But in those cases, we have to write down the limitations, that these are the limitations of the analysis. And also we'll have to basically conduct a sensitivity analysis by removing one set of studies. Either we will do with only the RCTs, and then we will compare the results of the base case, that is the first analysis, and then we will redo the analysis, removing a certain set of studies that might be observational studies in this case, and redo the analysis, and then both the results are compared and reported. Then we check for the statistical heterogeneity. It is basically based upon variability in the intervention effects being evaluated in different studies. So in an ideal scenario, all the studies should be of a similar time frame also. Suppose if there is a drug A and we have conducted a clinical trial for that for 6 months only, and for a drug B, the clinical trial is there for 2 years. So 6 month data cannot be compared with 2 years data. It should be similar in terms of variability, means in terms of effect outcomes as well, also the time frame. So after judging this, we prepare a network diagram. Those generally look like this only. First one is a single closed loop. You can find these as well. These are basically a treatment, and these lines means there are clinical trials comparing these. We tend to write the clinical trial name as well. Suppose there is a trial ABC comparing ibuprofen versus indomethacin. They will create a line here and write the trial name also. And the width of these lines means the number of clinical trials comparing these two treatments. So the width will increase as the number of trials increases. Similarly, we have a star network and then a connected network and a complex network. This is purely based upon the studies included. We cannot basically assess or we cannot judge that which type of network diagram will come out. It is purely based upon the studies that are included in the systematic literature review. Now, I was talking about heterogeneities. Now, actually, how to check for those heterogeneities? We prepare certain tables where we compare certain data for the included studies, like what is the publication type, in which countries those are conducted, because in certain scenarios and certain disease conditions, we cannot compare Indian data with the US data because the patient demographics are quite different in these areas. Then we also check for the number of patients included. A study with 10 patients cannot be compared with the study of 3,000 patients. So it should be similar across. Then what is the intervention and comparator? Why we check for these? We check for the doses. Like here for amisulpride, in one trial it is 400 to 800 mg. In the next one it is 200 to 800, and it may vary as well, and it is varying across the clinical trials. Similarly, we also check for the comparators, that what is the dose given to the patients. Then if phase is reported, then population, if there are adults, adolescents, geriatric patients, or only females or males, we also populate that. Then what is the population type? How the patients were included in the clinical trial, that is there in the systematic literature review. Whether it was an inpatient or outpatient study. Inpatient means the patients that are admitted to the hospital, and outpatient, those are taking medications at their home. This is basically to know that what type of patient structure we have, because yes, there are cases of compliance in terms of patients. Suppose in a hospital, if you have to take a certain medication 4 times a day, nurse will come and it will give you on the fixed time 4 times a day that medication. But when you are taking the medication on your own, you might skip one, or you have basically the gap between the two medications or the two doses might increase or decrease because you are working somewhere, you are traveling. So these type of compliance issues, that's why we capture this as well. Then what is the trial design? Like a single center, prospective, double blind clinical trial. So a similar set of studies can be compared. And this table we prepare, any of the decision to include or exclude a study from a network diagram or a network meta-analysis cannot be taken by the person who is doing a network meta-analysis. These decisions, suppose this study from Taiwan needs to be excluded based upon any criteria, this decision will be taken by a KOL. KOL is the key opinion leader, which are generally the renowned person in that treatment line. Suppose if it is of oncology, like this one is for schizophrenia. So it should be a renowned doctor from that area only. And that renowned person or KOL has to be bring from the client side and should not be from client's team. So these are generally basically a common people, so that we should not introduce biasness here. These decisions should be purely based upon their clinical practice and what they see in the market while treating patients. So we prepare these type of tables and then we show this data to a KOL. Yes, client is also available in that calls. These are generally 1 to 3 hours discussions with KOLs, where we take all the decisions which study needs to be included or excluded. A person who is conducting a network meta-analysis, in an ideal scenario, when you're working with the clients, pharmaceutical clients, these decisions should not come from your side. These decisions should come from a KOL. But yes, when we do these type of assessments or these type of analysis in college, we tend to take help of our professors as well. Then we also compare the quality assessment. This is a chart from risk of bias assessment that is done for the studies reporting on randomized clinical trials to judge that whether all the studies of higher quality, and if there is a study which is having a very bad quality or the bad higher risk of biasness, that should ideally be excluded from the analysis. And then we also show for the individual studies that which studies are having higher risk of bias and on what parameters. This is shown to the KOL, and they take decision whether to include in this case or not. And whenever we are showing these type of for the highlighted studies, like which are having reds in there because of high risk of bias, we tend to open the PDF of these studies because KOL can ask, can I see that publication? So always be ready with these type of discussions. Then we check for the transitivity assessment. These are the effect modifiers that we were discussing in the previous slide. As this is for schizophrenia, some of them would be specific to a disease area, but some are general ones, like whether the clinical trial conducted an inpatient or outpatient studies, what is the age, what is the gender, race and ethnicity. So these are the general baseline characteristics of the patients that are enrolled in those clinical studies. Then we come age of onset of specific disease duration, because if a patient of diabetes, if he or she is taking the diabetic medication since last 20 years, and a newly diagnosed patients, the response of the same dose would be different in both the conditions, both the patients. So we also check for disease duration, age at which the disease has occurred, what is the weight of the patient, why weight here? Because with schizophrenia treatment, there is a side effect of weight loss. That is why we have checked the weight as well. Because if the a patient pool is there with the average mean patient weight is 20, that cannot be compared with the average mean weight of 80 kgs in another study. Then we check for disease specific, like here we have PANSS score, PANSS score, that's total score, negative symptoms for positive symptoms, what is brief psychiatric rating scale, then there's clinical global impression. These vary with the disease areas, and this come from the baseline characteristics of each trial. Then how to show this to basically to our KOLs or doctors so that they can take decisions. So we prepare these type of histograms. This is called a histogram, and this for each bar is for a specific treatment in each trial. These are amisulpride trials. For the amisulpride clinical trials for the treatment arms, what is the mean age? Here you can see that there are two to three trials or two to three treatment arms which are having patient mean age between 15 to 16, whereas others are above 30 or so. So ideally, these should be excluded from the analysis. So that the KOLs can take easy decisions, we prepare these type of histograms and club them by treatments, not by the clinical studies. And if you have a limited number of studies, 40, 50 studies that are included in your SLR, then those type of histograms are fine. But what if you have 200 to 300 studies? Then you cannot show that in a histogram. Why? Because it will go from basically more than 10 to 12 slides. So a KOL cannot go through all the 12 slides and take a decision because the data will not be visible or visible. Also, he will not be able to judge accordingly. So in those cases, we prepare this box plot. Here, each box and plot is basically referring to a treatment, which is reported in multiple studies. Now, how to understand that box plot? So the center line is the median value of the reported mean age across all the trials of a treatment. Like for amisulpride, this if there are 10 trials, this is the median value. This can be easily prepared in any software as well as Excel as also. What is the upper quartile and lower quartile of the same? These are the IQR ranges. And then what is the maximum value that is reported among all the clinical trials of that treatment? And what is the minimum value? Now, if you go here, there are certain trials which are reporting below 20 as well. Then we will highlight these specific studies in a separate histogram that which one are these studies and whether they should be included or excluded. That decision will be taken by a KOL based upon the clinical practice. So this is basically what we do in a feasibility assessment, and then we present this data to a KOL and we ask them to take decisions. Now we will understand what are the outcomes. I might be going too fast because I have to cover a lot of things in this session only. So you will have the recordings with you. You can go through that again. And I will be there to answer all the questions in the end. Now, there are two types of outcomes, binary or dichotomous. These are the same, and there is a continuous outcome. Binary or dichotomous outcomes basically when the outcome for every participant is one of the two possibilities. If you're working for a oncology trial, you have oncology data and mortality is one of the outcomes. Either the patient will die or will not die, will be alive. So in these type of scenarios, either the patient got treated or not got treated, whether a side effect has occurred or not occurred. These type of events are reported in terms of binary outcomes because you have binary data. Whether the event has occurred means 1, whether the event has not occurred means 0. And these are usually reported as risk ratios, odds ratios, risk differences, number needed to treat. And suppose these are basically n by n, small n by capital N. Capital N is the total number of patients in that treatment arm. By and the numerator is basically small n, that is the in how many patients that event has occurred. Like suppose here we have an example of gastric ulcer healing. If we have taken a patient with gastric ulcers, given treatment vanoprazan, and to other set we have given placebo. So 50 out of 80 patients had healing at that specific time. That might be 6 months, 6 weeks, for what time that clinical trial has been conducted. So it will be 50 by 80. 80 were the total number of patients enrolled, and 50 has gained gastric ulcer healing. Similarly, for placebo, if 80% patients were enrolled, 20 has gained that. So this is reported by n or percentages. So these are the binary outcomes. Now, what is risk and odd? So in our pharma world or in our basically clinical practice terms, we think that these are interchangeable, but these are not an interchangeable words. Risk ratio and odds ratio are quite different to each other. Let me have, yes. So here, how to calculate a odds ratio. So odds is basically the odds, it is the ratio of odds of having that event occurred in that treatment arm. There were total 50 events in 80 patients, the same example. Now number of events, no events is in 30 patients. So we will be dividing 50 by 80, and this is the odds of having that event. No, we will be dividing by 50 by 30, event divided by no event. How many event has occurred in those patients divided by how many patients were there in which that has not occurred. We will be calculating that, and then we will be dividing it by, and both the odds we will be dividing, we'll calculating the ratio. Ratio is reported like 5.03. It means it's the having the ratio of having odds of that event vanoprazan versus placebo, upper versus lower. This is reported like this. And risk we generally divide by event by total number, and then we calculate the risk ratio. Odds, whenever we are calculating odds ratio, it is that how many had that event divided by no event. Suppose we have example of having rashes, that's a side effect. 20 patients had the rashes, while in a pool of 80 patients, means 20 divided by 60, 60 will not have that rash. That will be the odds. For the risk, it is 20 by 80. This is how a binary or dichotomous outcome is reported. Now we come to continuous outcome. These are outcomes basically, it happens where change happens in due course of time. Suppose we started a clinical study in diabetic patients. HbA1c level at the baseline was 10.2. After due course of treatment for 6 months, now the mean HbA1c level of the patients in treatment 8 arm has reduced to 8.2. So the reduction between them, 10.2 minus 8.2 is 2. 2 is your mean change from baseline. That is reported in terms of mean change from baseline, because there is a continuous change that is happening. It is not a single event that whether rash has happened or not, but yes, there's a continuous change happening in the, so these are reported as dichotomous outcomes. Why we are discussing this? Because whenever we conduct analysis, the codes that we use for binary as and the continuous outcomes are different, because the structure of data is different among these. Now, how, like I have given the example here, we have example of reduction in MADRS score in the patients. At the baseline, it was 25, and then there's a reduction. In 6 weeks, it reduced to 15. Change from baseline, 25 minus 15, that is 10. It is reported like this. Either it will be reported a baseline and endpoint, or it is reported as change from baseline. In the treatment B, at the mean baseline was 25. After treatment for 6 weeks, it has reduced to 20. Change from baseline is 5. It means that treatment A is offering 5 point more reduction with the treatment, means it is A is better than B in reduction of MADRS in a specific set of patients. This is how we report a continuous outcome. Now you have done the feasibility assessment, and you have also identified which type of outcomes are there, and you will now move ahead with the analysis. Prior to doing the analysis, there are certain values that are reported different in different trials. Suppose there is a trial which is reporting as mean change and standard deviation. Another trial is reporting as mean change and standard error. Third trial is reported mean change and p-value. Fourth is reporting mean change and 95% CI, that is confidence interval. Now how we do the analysis? Then we recalculate these variabilities or the baseline and endpoint values or mean change as per our requirements, and then after that we do the analysis. These formulas you can find in Cochrane Handbook for Conducting Meta-Analysis. These are not basically developed by me. These were suggested by Cochrane and vetted very well. You can use this. If standard error is not reported, and in the trial we are reporting 95% CI value for a specific set of patients, then standard error can be calculated by subtracting 95% CI minus 95% CI lower divided by 2 into 1.96. This is purely a mathematical formula. You can directly use it. If standard error is not reported or a standard deviation is reported, then to calculate standard error, standard deviation divided by square root of number of participants in that treatment arm. And if you need SD, similarly, you can multiply standard error with square root of number of participants in that treatment arm. What if standard deviation at baseline and endpoint is reported and not a change for change it is not reported? Like we have calculated the mean change from baseline by subtracting the final outcome value by the baseline value. Standard error cannot be calculated like that because this is the variability of the data. So standard error of the change, if standard deviation at baseline and endpoint is given, it is the complete square root of square of standard deviation at baseline plus square of standard deviation at final minus 2 into correlation SD at baseline multiplied by SD at final. Correlation value, it should be given in the clinical trials, but usually you not find this in the publications, but in the CSRs and other detailed documents, it is reported. But if it is not reported, you can assess it. No, you can use it at 0.5 divided by square root of sample size. You will find these formulas in Cochrane Handbook, so no need to take screenshots here. If standard error at baseline and endpoint is given, but standard error for change is not given, you will calculate it by complete square root of square of standard error at baseline plus square of standard error at endpoint minus 2 into correlation into SC baseline and SC endpoint. Correlation will remain same if it is reported in the trial, otherwise assume it to be 0.5. Okay, now we'll discuss about NMA. So can I take questions as well if there are? I cannot see that actually. Okay, I will take that in the end. So this is how we collate data. After finalization of the studies for the NMA, after basically removing the studies in which complete data is not reported, if it can be calculated, we can calculate. Otherwise, if basically suppose mean is given and we don't have any variability reported in that sometimes happens in conference abstracts, then we have to remove that study from our analysis because we need complete set for continuous outcome, mean and standard error, and in some codes mean and standard deviation. So either you will have a variability, then only you can include that study into the analysis. So suppose here we have study 236. It has 3 arms. Arm 1, that is of placebo. This is the mean change from baseline for MADRS change, and this is the standard error reported. For arm 2, that is lurasidone. This is the mean change from baseline for MADRS reported for 6 weeks and standard error. Then we have arm 3, that is also lurasidone. That is, and the data is like this, mean and standard error. Why 2 lurasidone arm? Because these had different doses. But the objective of NMA as well as systematic literature review, we were not doing dose separated analysis. We can do it like that. This is the resident 20 mg and resident 40 mg separately. But in this specific example, we were not doing dose separated analysis. Similarly, for other, like third example, say 2008 study, we have arm 1 placebo and arm 2 aripiprazole. Why we have put treatment arm 4? Because there is one study of cariprazine which has 4 treatment arms. Now we'll populate this type of data like this in Excel file, and post this we will create a file for doing the analysis. Now let me show you that how we do the analysis. So today we will be using R for this. For conducting the analysis, you should have R, R Studio or R Shiny based upon the codes that are you using. And in the background, you will be using OpenWorks or WinWorks. There are various codes that don't require OpenWorks and WinWorks as well, but that completely depends upon the codes that you are using. And these codes are freely available on Google. You can search for R code for conducting network meta-analysis. You will have ton of them. Use any of that and do the analysis. There are free softwares like RevMan and other as well, which can be used for conducting network meta-analysis. Prior to this, we used Stata, and then we were using OpenWorks and WinWorks manually. But in the recent years, in last 5-7 years, we have started using R, through which we conduct analysis on OpenWorks or WinWorks, because this is quite easy. Otherwise, in WinWorks, you have to put a lot of manual inputs and commands. And if you miss anyone or you have put it wrong, then you'll have to do the analysis again. And it was very time consuming process. That's why we have shifted to using R for basically ease of doing the analysis. Generally, we have a fixed effect and random effect models. Generally, we use random effect models for reporting the results, because the results are more streamlined in this. But in any case, if you are reporting network meta or meta-analysis results, you will have to report both the outcomes. Suppose for any specific outcome, you are running fixed effect and random effect. But for random effect, the results are better. You will show or basically report random effect results in the main body of the manuscript or whatever wherever in the NMA report. But in the appendix, you will also have to report the fixed effect results. There are various parameters to judge that which outcome is doing better on which effects model. Based upon those, you can select your model for reporting in the main body as well as the appendix. But both the results should be reported in all the cases. You cannot miss that I have used fixed effect or random effect. Now, how the model looks, this is the R script that we will be running. Prior to R script, how we will input the data into a system. We can use Excel file as well, but let me show you. So the same data has been transformed to this and these notepad files. Now, first of all, we will give numbering to the treatments. How will we give? So treatment 1 is placebo, treatment 2 aripiprazole, treatment 3 cariprazine. You will give numbering to them, because this OpenWorks, WinWorks these are based upon binary languages. They don't understand words. They just understand numbers. So you will first give them the number for each treatment. And similarly, we'll create this data file. Like here you have write the mean change from baseline for treatment arm 1. Treatment arm 1 is 1. That was placebo as per the numbering given to it. Like here, the 7th number has been given to lurasidone, means in this first study 236, there were 2 lurasidone arms. Those are numbered as 7 and the data is reported like this. Whenever we are using R, anything written after hash, it doesn't recognize that. It is like that for our language. Anything written after hash is not for the analysis. It's just for the documentation purpose. We write study names here so that we can do the QC of the data as well and keep it basically the data is same and there is no error while populating these sheets. In most of the cases, you will see that people use Excel files to as an input file for these type of analysis. But what happens if this is the Excel file and there is a space minus 10.7 a space there? You will not be able to do the analysis because that's an error. And to identify that, we prepare these notepad files. Because here, if there is a space, so the alignment of the entire column will be disturbed. And you will be able to identify that there is an additional space there. Okay, these are the inputs that we generally give to R for conducting the analysis. Now we have our script that we will open in R Studio. So this is how a R Studio looks like. Basically, this is the console where when we hit any command, it will do the do as directed. First, if we have if we are starting, so like I have told you that if there is a hash prior to anything, it will not read that. First, we have to install certain packages like R2OpenWorks, RUtils for forest plot, for Coda, for ggplot to run a specific command, and you will find these codes available on Google. So these are freely published. You can use any of them. Whenever you do analysis in this R, if you selected something, you can click run from here, or shortcut for that is Control plus Enter. I have those basically codes with me, or the packages that are already installed in my system. If these are not installed, installation process will occur here for around 2 minutes, and these will be installed in your system. Then we will put command for controlling, for clearing the workspace. If any data was there already inputted, it will clean that. Then you will set the working directory. From where this is coming from that file only. So the working directory is location of the data where you have kept it. Because R cannot assume, R cannot automatically pick data. You will have to give a location. Yes, here were the input files that we have kept. It is C. In that C file, I have kept NMA folder, in NMA folder normal, and then FE, fixed effect. So we will update that here. And I have given a command, like you will find these commands as well, how to use. So just run this. Now, whether your outcome is a good outcome or a bad outcome. Good outcome means where there is a betterment with the increase of specific thing. And bad outcome is basically where there is a betterment with decrease of something. This is just for the reporting purpose. There is no good or bad in those senses. These are for the mathematical understandings only. Like if there is a reduction of HbA1c, means if it is reducing, that means it is better. That will be a bad outcome. Just to identify in mathematics, we have to tag them here. What happens if we tag it as a good outcome? And there's a reduction of -2 or -3 in one drug, where if there's a reduction of -4 in another, it will say that -2 is better, -3 is worse. But actually the -4 one is better because it is offering a greater reduction. To basically give the ranking to those treatments, we have to say that these are good or bad outcome for the analysis purpose. You can edit the endpoint name. You can edit time point as well. This will come in the forest loss. All other commands I will give in a single go. Then there is a niter and n burning. So what happens for this network meta-analysis? We just not do a single analysis. We do it 1 lakh times or 100K times. And these are basically simulation based models, means each analysis is conducted 1 lakh times. Like here the n iter is n number of iterations. This is short form for the iterations is 80,000, and n burning, it is 20,000. You can change it as well. If you are doing a set of analysis for just for running it, you can make it 8,000 and 2,000 as well. But in the ideal scenario provided by the guidelines of the mathematicians only, at least it should be 80,000 and 20,000. It means the analysis will run for 80,000 times, but the model will basically burn or will not take into considerations the initial 20,000 iterations. Why? Because at that time, model adapts to the data. There might be a large variations in those data sets or the reporting data sets. So to adjust to the model, or to basically what we say that to basically adjust the model or let the model sit down, if we call it in layman language, we burn initial certain iterations and we use the later ones. So here we'll be doing the analysis for 80,000 times, but removing the initial 20,000 simulations. and we'll be using another 60,000 for the reporting. Then we'll load the data. These are general commands. It will call OpenWorks. It will open OpenWorks in the background like that. So let me do this. So you can do multiple commands in a single go as well. So the data has been inputted. If you have to check that whether your data is not going. You can go up. Here you can see that your data has been inputted from that notepad file. If you see a variation that this 3 is coming here, it means there is error in the data. If you see that if it is blank here, means there is an error in the data. If there is number of arms is 3 and you see basically a treatment name here 7 as well, then it will shoot a error because in the corresponding columns it is NA. Similarly, the treatment numbers that you have given in that notepad file, it will take that. So you can check your data from here. Now we will call OpenWorks to do the analysis. This generally take 2 to 3 minutes. So let me find out questions by the time if there are any. There are no questions at the moment. So I would advise if you have any questions till now, you can put them in the chat box and I will answer that. So once this analysis is conducted, then we will export the results into the Excel file as well as rankograms and meaning of MADRS score. And it is MADRS. Full form you can check on Google. This is for reporting the basically severity of disease, that is schizophrenia. This is a scale basically. MADRS scale, there are certain questions in it which we ask to the patient, and based on the response of the patients, we give certain ratings, and that way we check for the severity of schizophrenia as well as other psychiatric disorders as well. Okay, let this run, and if you have any other questions, you can take it. This MADRS is just an outcome from that clinical study. It can be overall survival, progression-free survival for oncology, side effects, for hypertension, it will be the reduction of blood pressure, and for diabetic, it would be the reduction in fasting plasma glucose or a reduction in HbA1c. So it is based upon the treatment and the indication for which we are judging that treatment. Okay. So if whenever you are working on any R, basically R software, if it is red here, don't click anywhere on R. It means it is doing the analysis or it is following your commands and doing the things. Don't interrupt it in between, otherwise it will hang and you will not get the response. Any questions till now? It was simple or it was too tough that you didn't understand a word? You can respond in the chat message as well. Were you able to follow the session? No responses yet. Okay. So you need not to calculate risk factor in binary. It will be reported like that in terms of odds ratio or risk ratio. You have to take that from the publication of the clinical trial. Suppose you have a clinical data. Please slow down a little. Okay. So you will get this risk factors and everything from basically risk ratio and odds ratio reported in the clinical trial. You will have to extract that from that. Can you please mention which course we need to do for R or R Studio for anime? First of all, do basic from LinkedIn Learning, basics of R. There's a free course available there so that you come to know that how to give commands and how to use those codes, free codes that are available online. Then you need not to do any other thing. If you can run R, you can run this code and it will work. Okay, we are following, but the topic is new facing the, yes. So it's a completely mathematical topic, not related to pharma or those clinical sides. But you will have the recordings. You can go back anytime, listen to the recordings, and then you will able to understand more. Also, you can use AI as well as Google Forums or Wikipedia to understand the terminologies that I have used. Learning meta-analysis or network meta-analysis cannot occur in a single session because that's a mathematical. You will have to do the practice and understand everything multiple times, like we used to do equations in our schooling, and some people might have done that in their graduations as well. You will have to practice mathematics. Likewise, you have to do this analysis again and again so that you come to know about each aspect. Now our analysis is done. Next command is exporting results in Excel, preparing tables, then creating rankogram, ranking the treatment tables. We will develop forest plots. These are the just codes that were there. We will just run them. Once you run this, it will take time, but let me show you where the results will come. So in the results section, it will start populating results like this. As it is going on, we cannot open it, but I have these results in a slide. We'll show it from there. Now, this was the anime input, how to do the analysis. We have gone through that. Now, what are the anime results or anime outputs? So the majority of the, basically the major thing in an anime for a clinician or for a pharmacy people or for who are working in HEOR is basically to understand the feasibility assessment in detail, how to identify an outlier study, whether in terms of age or any other effect modifier, prepare data tables like I've shown in the previous slide, and there are biostat people that are available to run the analysis in each team. But then they give you crude results like this. Then you have to understand this, and based upon this, you have to write the findings in anime report, in your publications, in the slide deck that you present to the clients. So the most important thing is the feasibility assessment and understanding the results. Running a network meta-analysis or running the analysis, you can take help of biostat people. They can do it very easily. Now, this is the first outcome that you see. It is in terms of forest plots. Now, to give you a break, so why we do a network meta-analysis? So that you can digest the things that I have told you till now. What happens in late 1960s and 70s, there were concerns from FDA. For the approval, they wanted clinical trials for each drug. After the thalidomide tragedy and all those things happened in 1960s and 70s, then they shifted that for each approval, you need a phase 3 clinical trials of this number of patients in this indication. There were strict guidelines. But when this moved on to 1980s and 90s, there was a need of an analysis that can bypass a clinical trial. Suppose I am coming into the market, I cannot do 5 clinical trials comparing my treatment with the standard of care, with placebo, with other competitors. But to showcase that my drug is equally or more efficacious than the competitors and placebo, I needed, or basically the pharma community or the pharmaceutical companies, they needed analysis. Then this network meta-analysis came. Suppose my drug is drug A and Richard's drug is drug B. I have done a clinical trial comparing drug A versus C. Richa has done drug B versus C, but there is no direct trial comparing A versus B. So for having the value or the comparison of each outcome for these A versus B, we can conduct a network meta-analysis. That saves billions of dollars that pharma companies have to put for conducting multiple phase 3 trials. Generally, nowadays, pharma companies conduct 1 to 2 or 3 max phase 3 clinical trials for their molecules for a single indication. But earlier that time, although the treatment options were very less, but as the treatment options were increasing, these direct comparisons were taking a lot of cost. That is why this network meta-analysis or meta-analysis came into existence in late 90s. Then the outcome was this forest plot. Not this forest plot, actually, this league table. But when pharmaceutical companies present this to FDA, they know this, but when they use this type of data for their promotions, KOLs or physicians, they don't understand this type of mathematical data, that what they want to say with this. So to ease this process, we came up with forest plots. How to read this? First comparison is aripiprazole versus placebo. And here there is a chart, favors intervention or favors comparator based upon good and bad outcome, basically positive and negative outcome. How to read this? Aripiprazole versus placebo is favoring intervention, means aripiprazole. Aripiprazole is offering 1.09 more change in baseline, more change from baseline MADRS as compared to placebo. If placebo was offering reduction of 10, aripiprazole was offering 11.09, means 1.09 more reduction in this specific outcome versus placebo. And these are the 95% CRI. You will have seen CI, that is confidence intervals, earlier. But whenever we report results in form of which has been calculated through simulations, multiple simulations, more than 1,000 simulations in any case, this 95% CI is said as 95% credible interval, not confidence interval. It means that 95% credible interval is ranging from minus 3.24 to 1.08. This is your effect. This is 95% CI. And line at 0, in case of binary outcome, this line will be at 1. This line of 0 is a line of no effect or null effect. Means if this center box is at this, mean the difference is 0. There is no difference in comparator versus intervention. So this is your effect. Like we take another example, caripiprazole versus placebo. Cariprazine is offering more reduction by 2.40 versus placebo for change from baseline in MADRS, means it is better than. So this is basically to interpret, to show that which drug is better than which one, which comparison. We can likewise have the analysis of lurasidone versus aripiprazole. Lurasidone is offering more reduction in MADRS as compared to aripiprazole because this is here. And whenever a 95% CI value, this bar, it crosses the line of null effect or no effect, it means results are not clinically significant or clinically meaningful. We used to write as significant, but here we write as meaningful. Why? Because the data is coming from multiple simulations. We cannot write it statistically significant, but we can write it as statistically meaningful or clinically meaningful. So these are some separate terms that are from the stat thing or the statics that we use whenever we are reporting any data from simulation, more than 1,000 iterations. These terms are changed, like confidence interval is changed to credible intervals. Statistically significant is changed to statistically meaningful, because this is not coming from a single analysis, but a simulation of multiple analysis. Any questions on forest plot? You can shoot. Now, that forest plot is prepared from this type of data. This is the primary outcome that we get from network meta-analysis. And this will be there in that folder, which I have shown. You will already get results there. Here, certain values are bold. And how to read this? Like we had aripiprazole versus placebo. This was the value minus 1.09, minus 3.24 to 1.08 as 95% credible interval. That forest plot is populated based upon this data, aripiprazole versus placebo. Like you have seen this for the cariprazine versus placebo, the 95% CI value is not. What is the range to know if the outcome is good or no? There is no range for that basically. That is comparison of the data based upon that. So for this comparison, caripiprazole versus placebo, the 95% credible interval line is not crossing zero. That means it is statistically meaningful. And similarly, in that also, we have made it bold so that we could identify easily which are the values that are statistically meaningful and which are not. The non-bold has crossed line of zero, like it has started from -3 and reached 1. It has crossed zero. But for this, it has started from -3.54 and on the same side in -1.23, it ended the credible interval. So these are basically read like this. Aripiprazole versus placebo is this. If you want placebo versus aripiprazole, this would be the value. Here, if you want to compare placebo versus aripiprazole, you will read this data. This is called as league table. Earlier graph was called as forest plot. Now what happened? Even reading this forest plot, which came into existence, so the physicians were still not that happy, or they were not able to interpret these forest plots. And generally, the marketing people that take this data to the KOLs, physicians, doctors, they were not able to explain this to the doctors. So this I'm talking about when we are doing medical marketing. So then they came up with rankograms. This is a rankogram and it is based upon the Sukra values that you might have read in late schooling. Sukra is the surface under the curve area. So whatever the surface, the area behind this curve is, it is called as Sukra. These are the Sukra ratings. Based upon that, we have the Sukra rankings. First of all, let's understand this. Higher the Sukra, better the treatment outcome. Like here, we have the highest one is quetiapine, 0.92. Next, second ranking is olanzapine, 0.87. Based upon the Sukra ranking, we rank the treatment which is best for that specific outcome, not for the overall treatment. If these analysis we do for specific outcomes, and the results are reported in terms of each outcome. First one is quetiapine, second is olanzapine. We tend to report the data in this manner. And how to read this graph? This is probability versus ranking. Probability of having that molecule at that rank. For the first ranking, whichever is the highest, whichever has the highest probability will be at top. Like for the first ranking, it is blue one, that is quetiapine. For the second ranking, it is olanzapine, that is green one here. Similarly, placebo, which is rated last because it has the lowest Sukra value for this outcome, will be ranking highest at 7 ranking, this here. This is how we read a rankogram. This is how we conduct a network meta-analysis. No questions, Now what happened in pharma world around 10 years back, physicians again raised that, okay, it's fine that you have this rankograms, you have forest plots, we can understand them better now. But what is the quality of this data in this league table? Because each value is coming through various analysis, comparing multiple clinical trials. How we can say that each clinical trial is basically from a high quality, and what is the quality of these reported numbers? Then there was a society developed, or basically people joined, and they prepared guidelines. A similar one, which we have risk of bias assessment for randomized clinical trials. You will have black and down checklist for observational studies. There is nice checklist for appraising the publications of randomized clinical trials, Drummond checklist for economic studies, and like this, many more. Similarly, there is a grade assessment that is developed for assessing each and every value that is coming from a network meta-analysis. So we'll be doing the quality assessment of all these values, each value basically. For Racidon versus placebo, this is minus 3.49. This is 95% CI value or the credible interval value. We will assess the quality of this based upon certain guidelines. Yes, there are guidelines. So the grades is the grades of recommendation, assessment, development, and evaluation working group. That is called a grade working group. It has developed this guideline. What this guideline work, it basically check for quality of a body of evidence of each number, that calculations or the results that were there involves consideration within high risk of bias methodology. So it checked for methodological quality, what was the risk of bias of those individual publications, directness of evidence, whether the initial value is coming directly from a clinical trial or it is calculated ones. Then on heterogeneity, precision of effect estimates, and risk of publication bias. These 5 parameters, we check each value on these, and then we give it a basically of what quality that is. And this is a mandate nowadays. If you are going for a publication, if in your manuscript or in the appendix of your manuscript, you don't have a publication or they don't have done the grade assessment, the manuscript, they will return it back with comments that please submit grade assessment as well. So after checking for the quality, we have four criteria, that whether the quality or the rating of that data point is high, moderate, low, or very low. Now, how to check for this? We have 5 parameters. We start with high, like we have here high, moderate, low, and very low. For each value, first we will give that value for each value like minus 3.49 with credible interval values, we will give it a high quality tag. Then we will check on these 5 parameters, and if that value or the input from which clinical trials is coming, if it is hampering or any of the parameters, we will downgrading it by 1 level. We will checking for all 5 parameters. Suppose the first one is limitation in study design and execution. So the input value for that specific comparison, if it is coming from a clinical trial in which random sequence generation was not done, blending method was not followed, allocation concealment was not done, we will decrease it by 1 level from high to moderate. Then we will check for the second one, that is inconsistency of results. We will check for whether the outcome assessor was blinded, whether there is an incomplete outcome data. How to identify an incomplete outcome data means, suppose in the methodology section of that publication of clinical trial ABC, in the methodology section or in the protocol that is sometimes available on clinicaltrials.gov, they have said that we will be analyzing 10 outcomes, but in publication they have reported only 8. That means they have deliberately not reported 2 outcomes, and the chance might be that their drug has not worked to that level or the outcome was not positive for them. You will identify it by, then there is, if there is an unexplained heterogeneity or selective reporting of outcomes, similar thing. So if any of this is happening with that clinical trial, we will decrease from moderate to low. Indirectness of evidence, for direct estimates, we will not decrease it by 1, but if there is an indirect estimates in the NMA results, indirect estimate means A versus B, there was no trial that has been calculated from B versus C and A versus C, then we will downgrade it by 1 value. Why? Because yes, we have conducted a network meta-analysis, but we have not done a clinical trial. That drug has not been tested for that specific combination or comparison in patients. That is why we are downgrading the NMA outcome there. Last one is imprecision. If confidence intervals are quite large, there is no definition for quite large, but in comparison to other treatment arms, if it is happening to a specific comparison, that means yes, there is basically the quality of data is lower. And if the results, or basically for any comparison, if that 95% credible interval line is crossing the line of null hypothesis, then we will decrease it by 1 level. Last one is publication bias. That happens in case of conference abstracts where we have very limited information, and sometimes we incorporate data from press releases, news releases that these pharma companies they. In those cases, there is publication bias. Why? Because we have very limited information related to the design of the trial, other outcomes. So they selectively report primary outcome. The primary outcome has been met, and the values were like this. This is for our treatment versus placebo or versus standard of care. Now, how to do this? We generally transform that league table into these tables, these type of tables, and then for each comparison, we'll check for the quality. Quality of direct evidence means the value that is coming from the data extraction file. For that, the Residon versus placebo, as we have direct this thing, and there is no limitation in terms of risk of bias, inconsistency, indirectness, we will keep it as high. We will check for all 5 parameters for the direct estimates which are coming from clinical trials, as well as for the NMA generated values. Like for this one, the Residon versus quetiapine, there is no head-to-head trials. That means there is indirectness of evidence. And for each calculated or basically for each step, you will put star marks or the specific marks that we have listed here. And if it is faltering on limitations and inconsistency, then we will basically put those marks, specific marks, so that the person who is doing it must be available, must be able to understand. These are not accurate, just a sample, because this is just for the training purpose, as we cannot say share the original data because that is client confidential. So this is how we judge the quality of each evidence or each value coming out of a network meta-analysis, and we report it like that. With this, I'm open for the questions. That's all from my side for today. And there is a book by Sophia Dyers and others on network meta-analysis for decision making. You can refer to this book for any of the issues or any of the problems that you might face. This is a very thick book, basically. Don't try to follow it and read it by page, but yes, whenever you are doing a network meta-analysis and you don't understand a specific topic or you are stuck somewhere, you are not able to run the analysis, whatever errors you are getting, go to the index, check for that specific page and that specific information. Apart from this, for your training purpose and reading, you can follow Cochrane Handbook for Conducting Meta-Analysis. That is available online. You can directly search for. With this, I am open to questions. Any question?
Any example you could give for like plotting in forest plot and any two drugs you can compare?
There are a lot of tools. Basically, you can use Revman, and generally the graph that I have shown, that is the forest plot, is auto-generated in R as well. You can just use the codes whenever you are picking NMA codes for R. It will auto-generate this, and we have used basically Revman as well. That is free for industry, academic institutes, I think. After Revman, we have used Stata as well. We have used OpenBugs and WinBugs, and then this forest plots from league table can be generated in Excel also. There are a lot of online tools available for this. So generally, the codes that we use for R, they have certain packages and commands that auto-generate these forest plots, league tables, and everything. Yes, Excel is also a good platform for forest plot. Yes. So initially, these were prepared in Excel only, but then in advancement of technology, now we have online tools and everything that can be used. There are various mathematical tools like Prism and all those, which can be also be utilized for preparing forest plots, box plots, all these things, and histograms as well. Any other question? You can unmute and ask as well. Okay. I will again ask, if there are any questions, you can put in chat box, or if you are able to unmute and ask, you are open to that. How and where we can learn R? There are online courses, a lot of online courses, free as well as paid. I would suggest doing LinkedIn learning course that is for R. You can search for introduction to R and these type of courses. You can learn from there. I would suggest first go for the free courses, and once you are well versed with the how to run commands and everything, and you can opt for paid courses at Coursera. There are a lot of platforms based upon your basically liking that which platform you want to use. What are the different plots we develop in NMA? First one is the league table that has the basic data. Then we create forest plots, then rankograms. These are the three main outcomes of a network meta-analysis. But for doing feasibility assessment, you have to prepare those histograms or graphs as well as box and plot graphs if you have multiple studies in your SLR. So these are basically the basic ones. And apart from this, sometimes we have to showcase data to client in a specific manner. Then we can prepare forest plots comparing multiple comparisons as well. Like the first plot you have seen, I have compared all the treatments versus placebo, then all the treatment versus lurasidone. So you can shape it as per your requirement and report the data in front of the client or in publication as you need. Any other questions? So we don't have any further questions. So with this, over to Dr. Richa Goyal and CleanMed team.
Thanks, Rajpal. Hi, everyone. I understand for some of you it's a new topic. So, what we encourage is to go through the recordings, maybe multiple times if needed, go through all the references and do a lot of self reading and self learning. That is how basically we also learned it. Practicing again and again. Rajpal will also be sharing assessment and quick quiz for you as a weekly activity for all of you. And with this, we end today's session. So thank you everyone for joining.
Thanks, everyone. Bye-bye.
Thank you. Bye-bye.
Thank you so much.
Bye.
Thank you, ma'am.
Bye-bye.